Hi students, myself Chandrasekhar, Zoology faculty from Sri Chaitanya. In Sri Chaitanya's NEET Sankalp 2.0 program, we are moving forward with one more important chapter in Zoology, that is human reproduction. So this is the important chapter because nearly 3 to 4, sometimes 5 questions they asked in NEET from this chapter, and that too some difficult questions, the questions related to the hormones and reproduction related questions they are asking. Let's discuss the important concepts within 1 hour and covering all the details of this chapter. Let's begin the chapter. See, as we are all aware that humans are sexually reproducing and viviparous organisms. They give birth to the young ones. Regarding the events of reproduction, see, the first event is gametogenesis. Production of gametes by primary sex organs like testis and ovary is said to be gametogenesis. Followed by insemination. The release of semen into the female reproductive tract called vagina is said to be insemination. Followed by fertilization. See, once the semen is released into the female tract, it will go to the fallopian tube. Ova from ovary also reach the fallopian tube. There the sperm and ovum is fused. That is said to be fertilization. The site of fertilization is fallopian tube. Next, implantation. Once the zygote is formed, it will form a particular embryo called blastocyst. Blastocyst will go and attach to the wall of uterus, that is endometrium of uterus. That process is called implantation. The site of implantation is uterus. Then the embryo grows further till the parturition in the uterus itself. That time period is called gestation, that is gestation site is also uterus. Followed by parturition. Giving birth to the child is called parturition, which is a neuroendocrinal mechanism. And then lactation. So lactation, after delivery, mother feeding the young with the milk is said to be lactation. So these are the events of reproduction, the important events of reproduction regarding the humans. Next, let's get into the topic male reproductive system. Regarding the male reproductive system, the male reproductive system consists of the parts like a pair of testes which are located in scrotal sacs. See this picture, pair of testes present in the scrotal sacs. The testes are located outside the abdomen in the scrotal sacs. The temperature maintained by the scrotum is nearly 2 to 2.5 degrees centigrade less than the body temperature, which is suitable for the synthesis of sperms by the testes. Next, each testis is covered by the membranes. The outermost membrane of the testis is called tunica vaginalis, followed by a membrane made up of dense fibrous tissue known as tunica albuginea. This albuginea forms a number of compartments in the testis. They are called testicular lobules. Each testis consists of nearly 250 compartments, and each testis the size wise 4 to 5 centimeters length and 2 to 3 centimeters in width. They are located in the scrotal sacs. Let us see the parts that are there inside the testis. See, the testicular lobules consist of highly convoluted tubules inside it. Suppose if it is a lobule, lobule consists of many tubes like this. These tubes are called seminiferous tubules. The seminiferous tubules are the sites for the synthesis of sperms. Let me discuss about seminiferous tubules first. After that, I will go for the parts that are related to the male reproductive system. See, this is the transverse section of testis showing the picture related to the seminiferous tubules. See, each seminiferous tubule is lined by 2 types of cells called spermatogonia, which are said to be the male germ cells, and serotoli cells. Function of spermatogonia is the production of sperms, that is spermatogenesis. Serotoli cells, they are lining the seminiferous tubules. They nourish the developing sperms, that means they nourish the male germ cells and also produce a hormone called inhibin. Between the seminiferous tubules in this space, interstitial space we can call it as a stroma. In this there are cells called interstitial cells, also known as Leydig cells. These Leydig cells, when they are stimulated by the luteinizing hormone, the Leydig cells produces group of hormones called androgens. The principal androgen is testosterone. Testosterone is responsible for the development of male secondary sexual characteristics like low pitch voice. It helps in spermatogenesis, aggressiveness, and it also controls the male sexual behavior. This is about seminiferous tubules. And in the interstitial species, along with the interstitial cells, they consist of blood vessels. They also consist of some immunocompetent cells regarding the protection of the testis. And these seminiferous tubules, once the sperms are synthesized in this, the sperms from seminiferous tubules, they will move into a network of capillaries known as rete testis. This is rete testis. So, sperms from seminiferous tubules are getting into the rete. Rete testis to the sperms gets into vasa efferentia. Many tubules that are arised from the rete testis, they will open into a part called epididymis. So, the rete testis is the connection between, epididymis is connected to the rete testis by small tubules called vasa efferentia. Regarding the location of epididymis, it is located on the posterior side of the testis, not in the testis, outside the testis. This epididymis, let me tell you the functions of epididymis. Epididymis temporarily stores the sperms. It nourishes the sperms temporarily, and the secretions of epididymis, they contribute semen, and those secretions are required for the maturation and motility of sperms. Then epididymis, it will continue as vas deferens. Vas deferens, see, it will come out moving upward from the scrotal sac into the abdominal cavity. See this is urinary bladder located in the lower abdomen. It moves into the abdominal cavity. There is a connection, canal between the abdomen and scrotum called inguinal canal through that. And only epididymis will not move. Epididymis along with some blood vessels, along with some muscles will get into the abdomen. That epididymis, muscles, blood vessels put together known as spermatic cord. So, what is this epididymis doing? See, after getting into the abdomen, it takes a curve around the urinary bladder. This is called seminal vesicle. From seminal vesicle, there is a duct is coming down. On each side, epididymis joins with the duct of seminal vesicle, forming ejaculatory duct. By passing through the prostate gland, ejaculatory duct opens into the urethra from both the sides. Two ejaculatory ducts. See, epididymis, it is involved in the conduction of sperms, semen towards the abdomen, takes curve around the urinary bladder, joining with the duct of seminal vesicle, opening into the urethra by passing through the prostate gland. This is regarding epididymis. And this is regarding vas deferens. Vas deferens opens into urethra as ejaculatory duct. Let me tell you about urethra. Urethra is formed from the lower end of the urinary bladder. There are parts like prostatic urethra covered by prostate gland. After that, membranous urethra. Then after that, the urethra passing through the male copulatory organ called penis, that is called penile urethra. So, what is the duty of this urethra? Urethra is lined by the transitional epithelium. The wall of urethra consists of smooth muscles. When urinary bladder contract, urine passage occurs through the urethra. But at the time of coitus, during ejaculation of semen, the semen will be released from ejaculatory duct into the urethra, and urethra conducts the passage of semen. So, the urethra in male humans acts as urinogenital canal. It conducts the passage of semen. It also conducts the passage of urine. The next part is penis. Penis is called male copulatory organ. The tip of the penis is called glans penis. The loose fold of skin at the tip of the penis is known as a foreskin. The duct that is passing through the penis is called urethra. And the penis consists of three longitudinal columns. The two columns are called corpora cavernosa. The ventral column is called corpus spongiosum. Urethra passes through the corpus spongiosum. On either sides, corpora cavernosa, which is made up of specialized tissues. Into that specialized tissue, there will be blood flow increase at the time of coitus. That makes the penis erectile. Penis is a erectile tissue. So, it is erectile organ. It is a copulatory organ. This is the organ involved in insemination, release of sperms into the female reproductive tract. Glans penis is the tip of the penis. The loose fold of skin at the tip of the penis is called foreskin. So this is about male reproductive organs. Now let's talk about male accessory reproductive glands. A pair of seminal vesicles present. One prostate gland is present. A pair of bulbourethral glands present. These are the accessory glands. And what about accessory ducts of male reproductive system? Seminiferous tubule is not accessory duct because it is involved in the synthesis of sperms. Then who are accessory after seminiferous? Rete testis, vas efferentia, epididymis, vas deferens, ejaculated duct, they are all coming under the male accessory reproductive ducts. Their function is to conduct the passage of semen. They transport and conduct, of course they also contribute for the formation of semen. So now let's talk about the glands, the accessory glands of male reproductive system. I said there are three types of accessory glands: pair of seminal vesicles, pair of prostate gland, one prostate gland and pair of bulbourethral glands. See this. Seminal vesicles, they secrete fluid which is alkaline in nature. It contributes to 60% of the semen. That is point number 1. The seminal fluid consists of fructose and prostaglandins. Fructose provides energy for the sperms. Prostaglandins, they increase the receptivity of cervix to sperm at the time of coitus when they are released into the female tract. One pair in nature, they are located towards the lower end of the, just below the urinary bladder. And below the seminal vesicles, a prostate gland is present. The secretions of prostate gland contribute 15 to 30% of the semen. And the prostatic secretions, they are involved in the activation and the motility of the sperms. Calcium ions are present in the secretions of prostate gland. And the third, below the prostate gland, a pair of bulbourethral glands present. They also secrete alkaline secretions. The functions of bulbourethral, the bulbourethral glands, they contribute for the semen, that means seminal plasma. Bulbourethral gland secretions acts as a flushing agent. See, if any urinary residues are present in the urethra, before the ejaculation of semen, the bulbourethral secretions can flush out all those urinary residues present in the urethra. Thereby, they will not let that urinary residues to damage the semen and sperms while it is passing through the urethra. Next, the secretions of bulbourethral glands, they also lubricate the penis. So this is about the male reproductive system. Regarding the male reproductive system, if you see the passage of a semen, that means sperms, first seminiferous tubules, then rete testis, vas efferentia, vas deferens, epididymis, vas deferens, ejaculated duct, urethra. From there, if it is the site of insemination, it will be released into the cervix, vagina of a female. Next, the secretions of seminal vesicles, NCERT point, secretions of prostate gland and bulbourethral glands put together called seminal plasma. The seminal plasma along with the sperms is called semen. So this is the important points related to the male reproductive system. Let's talk about female reproductive system. The female reproductive system consists of a pair of ovaries, oviducts, single uterus, cervix, one vagina and external genitalia located in the pelvic region, which is also called lower abdomen. See, the above parts along with the mammary glands are integrated structurally and functionally to support different processes like ovulation, fertilization, pregnancy, birth and child care. So the parts of the female reproductive system along with a pair of mammary glands, they structurally functionally integrated to support all these functions. I said they are located in the pelvic region, a pair of ovaries present. Let me show you the picture and explain this. See this picture. A pair of ovaries are present in the pelvic region. Ovaries are attached to the uterus. Ovaries are attached to the pelvic wall with the help of ligaments. Attached to the uterus with the help of ovarian ligaments, pelvic wall with the help of suspensory ligaments. And ovaries are held in position by a double-walled peritoneum known as mesovarium. And each ovary 2 to 4 centimeters in size. And each ovary consists of 4 different parts from outer to inner. the outer part of the ovary is covered by a germinal epithelium made up of simple cuboidal cells, followed by a dense layer protecting the ovary called tunica albuginea. This is tunica albuginea. The tunica albuginea is made up of dense fibrous. Tunica albuginea also present around testis, and followed by stroma. The stroma is made up of connective tissue. In the ovary, the stroma is divided into outer cortex and inner medulla. This is cortex and this is medulla. What is the function of the cortex? The cortex consists of different developmental stages of ovarian follicles. That means synthesis of ovum occurs in the cortex of the ovary. And what about medulla importance? Medulla consists of blood vessels, lymph vessels and nerves. So this is the basic structure of ovary, showing the parts like germinal epithelium, tunica albuginea, cortex and medulla in outer to inner order. Next, a pair of fallopian tubes present in female reproductive system. Each fallopian tube is around 10 to 12 centimeters in its length. Each fallopian tube is lined by simple columnar ciliated epithelium, and the wall of the fallopian tube consists of smooth muscles. With the help of smooth muscles and simple columnar ciliated epithelium, the fallopian tube can conduct the passage of ovum. When you see the parts of fallopian tube, it has three important parts: a funnel-like part called infundibulum, longest and widest part called ampulla, and a bridge that is connecting the fallopian tube to the uterus called isthmus. These are the three important parts. But at the end of the infundibulum, funnel-shaped infundibulum, there are finger-like projections present known as fimbriae. So infundibulum collects the ovum from the ovary with the help of fimbriae. And infundibulum is the part close to the ovary, whereas isthmus is the part close to the uterus. Infundibulum collects the ovum. Ovum finally reaches the ampulla. Sperms also reach the ampulla. Ampulla is the site of fertilization. Then the connection between the fallopian tube to the uterus is isthmus. Overall, the functions of fallopian tube is it conducts the passage of ovum. It is the site of fertilization. Next one is the uterus. Uterus is also called a WOMB, womb, metra. Uterus is also located in the pelvic region. The uterus is attached to the pelvic wall with the help of a peritoneum called mesometrium. Mesometrium. It has parts like fundus, body and cervix. The wall of uterus is having three layers from outer to inner. Outer layer is called perimetrium, middle is called myometrium, inner is called endometrium. Perimetrium is made up of squamous epithelium, myometrium is made up of smooth muscles, endometrium is glandular in nature. So overall, the uterus is muscular and glandular, inverted pear-shaped sac-like structure. And it is a distensible sac. While the embryo is growing inside the uterus, uterus expands in its size. And what are the functions of the uterus? Uterus, it provides a site for the implantation of the embryo. And in the uterus only embryo further shows its development called by a period called gestation. So it is the site of implantation, site of gestation. And in the uterine walls, endometrium is the wall which undergoes cyclical changes in menstrual cycle. It is the layer that sheds out during menstruation. It is the layer that will again synthesize in follicular phase. And about the myometrium, this is the layer that undergo contractions and relaxations. Actually, contractions of the uterus helps in parturition. Those contractions of myometrium are inhibited by progesterone. Those contractions are caused by the oxytocin. So this is about uterus. And then cervix. See, so the neck of the uterus is called cervix. It has cervical canal. Next part is vagina. Vagina plus cervical canal put together forming birth canal. Regarding vagina, till vagina they are all internal genitalia only. Regarding vagina, vagina is the site of insemination. So the sperms are released into the vagina. Vagina is a fibromuscular tube. It is lined by stratified squamous non-keratinized epithelium, and it is located between cervix and vestibule. Located between cervix and vestibule. These are the characters of vagina. Ovaries, fallopian tube, uterus, cervical canal, that means cervix, and vagina, they are all coming under internal genitalia. And what are the accessory ducts of female reproductive system other than ovary? Other than ovary, fallopian tube, uterus and vagina are known as accessory ducts of female reproductive system. The final part regarding the female reproductive system is external genitalia. Let's talk about external genitalia of female reproductive system. Regarding the external genitalia of a female reproductive system, there is a vestibule covered by two layers called wait one second. Yeah, labia majora, outer wall, and inner wall is called labia minora. So, into this gap there is an opening of urethra, the opening of vagina. Upper aperture is urethral opening, lower aperture is vaginal orifice. Above the vaginal orifice, that means above the urethral orifice, between two labia minora, there is a finger-like projection present known as the clitoris. Clitoris is also made up of corpora cavernosa. It is erectile in nature. Hence, it is said to be homologous to penis of a male. Above this, the skin with the pubic hair and fatty tissue present, collectively known as mons pubis. And the vaginal orifice either completely or partially lined by a mucous membrane called hymen. This hymen generally tear out during the first coitus, but hymen can also be easily removed by different activities. See, the hymen often torn during the first coitus. However, it can also be broken by sudden fall or jolt, insertion of a vaginal tampon, active participation in sports like horseback riding, cycling, etc. In some women, the hymen persists even after coitus. That is the reason why the presence or absence of hymen is not a reliable indicator of virginity or sexual experience of a female. So, regarding the external genitalia, labia majora, labia minora, clitoris, mons pubis. So, these are all coming under external genitalia. Till vagina, they are all internal genitalia. Next is mammary glands. A pair of mammary glands is present in female. Functional mammary glands are present in female. It is made up of two types of tissues. One is called adipose tissue. Second one is called glandular tissue. See, this one is the fat. Fat is nothing but adipose tissue. Remaining portion is glandular tissue. In the glandular tissue of a mammary gland, each breast consists of 15 to 20 mammary tubules. Mammary lobes, 15 to 20. Each mammary lobe consists of the clusters of cells for the secretion of milk. They are said to be alveoli. Clusters of cells are called alveoli. So, in the alveoli, milk is temporarily stored in the alveolar cavity. From there, the milk will be collected into the mammary tubules. Mammary tubules. From mammary tubules, it is collected into mammary ducts. Mammary ducts join to form mammary ampulla. Ampulla opens into lactiferous. Lactiferous duct opens at the tip of the nipple. So, regarding the passage of milk in mammary glands, first alveoli, alveoli to mammary tubule, mammary tubules to mammary duct, duct to ampulla, ampulla to lactiferous duct. Lactiferous duct opens at the tip of the nipple. So, the darkly stained area around the nipple is called areola. And about the muscles, see in this picture, they have given the impressions of ribs. Between the ribs, the muscles that are present are called intercostal muscles. In front of that, the muscles that are present into which these mammary glands are attached is said to be pectoralis major. So, this is about mammary glands. So, with this I have completed the female reproductive system. The next topic is gametogenesis. Production of gametes is called gametogenesis. In males it is called spermatogenesis. In females it is known as oogenesis. Let us talk about spermatogenesis point. Very, very important. The spermatogenesis starts from puberty due to the significant increase in a hormone called GnRH, gonadotropin releasing hormone of hypothalamus. In spermatogenesis, it is initiated by the cells called spermatogonia. also known as male germ cells. They are diploid in nature. They are located in the seminiferous tubules. The spermatogonia undergo mitosis first. So thereby the number of spermatogonia increases. That is called mitotic differentiation. Mitotic differentiation. See in this chart, I will show you the points related to the spermatogenesis. First, spermatogonia undergo mitotic division, and by mitotic division itself spermatogonia produces primary spermatocytes. So primary spermatocytes are produced from spermatogonia by mitosis. There is a slight difference in the size from spermatogonia to primary spermatocyte. Spermatogonia compared to the primary spermatocyte, they are smaller in size. Primary spermatocytes are somewhat large in size due to more amount of cytoplasm, and they also consist of more amount of DNA. There is no difference in the chromosomes. Both are diploid in nature. Both are diploid. Spermatogonia are diploid. Primary spermatocyte also diploid. See this? Then each primary spermatocyte undergo first meiotic division, meiosis 1, and producing a pair of secondary spermatocytes. As secondary spermatocytes are produced by meiosis, the secondary spermatocytes are haploid in nature. They are not having 2n, they are having n number of chromosomes, that is 23 chromosomes. Each secondary spermatocyte undergo meiosis 2, that is equal to mitosis, and producing 2 cells, each one producing 2 cells, they are called spermatids. Spermatids also haploid because they are produced by the meiosis 2, which is equal to mitosis. And finally, the spermatids by a process called differentiation, which is also known as spermiogenesis, they will be converted into spermatozoa. Here there is no division, only by differentiation one spermatid will become one sperm, one spermatozoon. So these are the steps. Spermatogonia undergo mitosis producing primary spermatocytes. Each primary spermatocyte is producing 2 secondary spermatocytes. Each secondary producing 2 spermatids. Each spermatid is producing one spermatozoon. In this final step, in the conversion of spermatid to spermatozoa, I said that process is called spermiogenesis. What happens in spermiogenesis? In the spermiogenesis step, differentiation step, the acrosome of the sperm is formed from the Golgi complex. Let me show you this picture. Acrosome of the sperm is formed from the Golgi complex. Next, the nucleus generally spherical, but it will be elongated in this differentiation process. The centrioles which are generally perpendicular, they will get separate in the differentiation process. Mitochondria will be arranged in the form of a spiral. So formation of mitochondrial spiral can also occur during differentiation. And axial filament extension through middle piece of the sperm into the tail can also occur during differentiation. So these are the steps that are there in spermiogenesis. See this formation of acrosome from Golgi, elongation of the nucleus, mitochondrial spiral formation, and also this axial filament formation. So this is about spermatogenesis. Human ejaculates nearly 200 to 300 million sperms. In the ejaculate of human, 200 to 300 million sperms present. 60% of these sperms should have normal size and shape. 40% of the 60% should have vigorous motility, which is required for the fertilization of the sperm. Fertilization of the sperm. So those numbers generally includes 200 to 300 total number, 60% is around 120 to 180 million should have normal size and shape, and 40% of this 60% includes around 48 to 72 millions, they should have vigorous motility. Secretions of epididymis, secretions of vas deferens, seminal vesicles and prostate, these 4 parts, they are responsible for the maturation and motility of the sperms. So with this I have completed the events related to the spermatogenesis, like how the sperms are formed, like haploid diploid stages, spermatogonia diploid, primary spermatocyte diploid, secondary spermatocyte, spermatid and spermatozoa are haploid in stage. So, what about the hormonal regulation of spermatogenesis? Regarding the hormonal regulation of spermatogenesis, during puberty, hypothalamus secretes GnRH. That GnRH will go and act on the cells of the testis. Acts on the adenohypophysis. Adenohypophysis produces gonadotropins, FSH, LH. FSH acts on Sertoli cells, produces certain factors. Those factors are involved in spermiogenesis. That is one point. What about the LH? LH acts on Leydig cells. The Leydig cells produces androgens. Okay, fine. Androgens are produced by the Leydig cells. When FSH acts on the Sertoli cells produce certain factors called androgen binding proteins. These androgen binding proteins and androgens are involved in regulating the spermatogenesis. At the time of stopping the spermatogenesis, the Sertoli cells produces inhibin. Inhibin will go and act on the adenohypophysis and inhibit the release of FSH. Thereby spermatogenesis will be regulated. So, regarding the regulation of spermatogenesis, hypothalamus produces GnRH. GnRH acts on adeno. FSH, LH are produced. FSH, Sertoli, androgen binding proteins. LH, Leydig cells, androgens, spermatogenesis. Inhibin produced by the Sertoli cells acts on adenohypophysis, inhibits the release of FSH, thereby regulates the spermatogenesis. So, with this spermatogenesis topic completed. Next, let us talk about oogenesis topic. There are remarkable differences between spermatogenesis and oogenesis. One difference is, the first difference I want to tell you, after that I will go for the other differences. Oogenesis starts before birth, but spermatogenesis starts from puberty onwards. So, what happens before birth regarding the oogenesis? During the fetal stage, listen carefully, during the fetal stage, the female germ cells, which are called ovum mother cells, also called oogonia, they start mitosis. They are present in the ovary, they start mitosis. In that mitosis, the oogonia number will increase. See, couple of millions of oogonia are formed in the ovary of a female fetus itself. It occurs before birth. Next, some of the oogonia are forming primary oocytes here. See this is primary oocyte. Before birth itself, primary oocyte starts meiosis 1, but arrested, that means stopped at prophase 1. So, formation of oogonia, formation of primary oocytes occur before birth itself. No oogonia formed and added after birth. Oogonia formation occur by mitotic differentiation. Even primary oocytes also formed by mitosis. But primary oocyte starts meiosis 1, but arrested at prophase 1. This is before birth. And before birth, one more event occurs, guys. That is, the primary oocyte is converted into a part called primary follicle. Primary oocyte develops into primary follicles. Very simple. Primary oocyte is a single cell. Around primary oocyte, thin layer of granulosa cells are formed. The thin granulosa along with the primary oocyte is known as a primary follicle. These primary follicles are also formed before birth. But from birth to puberty, what happens? I said couple of millions of oogonia. That means millions of primary follicles are formed. But from birth to puberty, majority of the primary follicles get degenerate by follicular atresia. At the time of puberty, each ovary of a girl consists of on an average 60,000 to 80,000 primary follicles. This is primary follicle. And from puberty onwards, there is a menstrual cycle. So, in the menstrual cycle, from puberty onwards, what will happen? Let me explain. So, the primary follicle during the follicular phase of menstrual cycle, it develops into secondary follicle. What is the difference between primary follicle to secondary follicle? Primary follicle is having primary oocyte with granulosa thin. Secondary follicle is also having primary oocyte, but the changes are the thin granulosa become thick granulosa here and also forming a layer called theca. So, theca for the first time seen in the secondary follicle. Then the secondary follicle develops into tertiary follicle. So, what are the differences from secondary to tertiary follicle? The difference is between the granulosa cells, there is a split at one end of the follicle and it forms a cavity filled with fluid. That cavity is called antrum. So, antrum for the first time seen in tertiary follicle. Along with that, the theca is organized, split into two, theca externa and theca interna in the tertiary follicle. Theca for the first time in secondary, it is organized to externa interna in the tertiary follicle. Antrum formation also in the tertiary follicle. Still, what is the oocyte in the tertiary follicle? Primary oocyte. Listen carefully, guys. This is primary oocyte which started meiosis 1 but stopped at prophase 1, 2n in nature. Now, it will complete the meiosis 1 in the tertiary follicle itself, forms two cells, one big cell and one small cell. This large cell is called secondary oocyte. This small cell is called first polar body. First polar body. Both are n in nature. There is no difference in the chromosome number between polar body and secondary oocyte. Both are having 23 chromosomes like 22 autosomes X, 1 allosome, sorry, A, 1 X, 23. 22 autosomes A, 1 X, 23. But where is the difference lies? In the amount of cytoplasm. So, as secondary oocyte is receiving more amount of cytoplasm, it has got some advantage like nutritional advantage, so that it can able to survive for a longer period when compared to the first polar body. And where is this completion occurs? This is completed in the tertiary follicle itself. Now, some more changes can occur in the tertiary follicle and finally it develops into a mature follicle called Graafian follicle. So, what are those differences here? See, the secondary oocyte with first polar body is present inside Graafian follicle now, and immediately it is surrounded by a layer called zona pellucida. This is a glycoprotein layer produced by the secondary oocyte. Then zona pellucida is surrounded by one more layer made up of granulosa cells called corona radiata. See, secondary oocyte, this one. First polar body, this one. This is zona, this is corona radiata. And between the secondary oocyte and zona pellucida, this space is called perivitelline space. Perivitelline space. Externally corona, after that zona, after that perivitelline space, after that the membrane of secondary oocyte is, which is nothing but plasma membrane. And other parts remain same, antrum will remain same, like tunica externa interna, but size wise compared to the tertiary follicle, Graafian follicle is larger. The Graafian follicle is also known as mature follicle. Okay, fine. Once the mature follicle is formed, with the influence of LH surge, the high levels of LH hormone, the Graafian follicle will be ruptured. When it is ruptured, secondary oocyte is released. This is secondary with first polar body, with zona, with corona and some granulosa cells. This total structure comes out by a process called ovulation. Actually, it is secondary oocyte, but commonly known as the secondary oocyte stage of ovum. Both are correct. So, by ovulation secondary oocyte is released. It can be known as ovum because it is the stage of the ovum by a process called ovulation. After ovulation, once the secondary oocyte and some contents comes out from the Graafian follicle, the remaining cells of the Graafian follicle develops into a temporary endocrine gland known as a corpus luteum, and it secretes some hormones like progesterone, estrogen, inhibin, relaxin. Once the secondary oocyte is released, first it is from the ovary it is released into the body cavity, from there fimbriae will collect it, from fimbriae infundibulum and reaches the ampulla of the fallopian tube. So, once it reaches the ampulla, there if it in contact with the sperm, sperm enter into the secondary oocyte. Once sperm enter the secondary oocyte now completes meiosis II. Actually, immediately after meiosis I, once secondary oocyte is formed, it will start meiosis II, guys, but it will be arrested at metaphase II. Now the secondary oocyte after ovulation, after the entry of sperm, it divides by meiosis II, produces two cells. One is ovum, which is also called ooted. The next one is called second polar body. First polar body is produced after first meiotic division. Second polar body is produced after second meiotic division. Secondary oocyte is produced after first meiotic division. Ooted, which is also called ovum, is produced after second meiotic division, that too after the entry of sperm into the secondary oocyte. Otherwise, meiosis II will not be completed until and unless the sperm enter into the secondary oocyte. Here the point I want to convey here, after secondary oocyte, I said ooted is formed, ooted itself is called ovum. That means there is no differentiation like spermatid to sperm conversion. There are many events can occur. Here ooted to ovum conversion, there are no many events, there is no differentiation. So differentiation is absent in oogenesis, whereas differentiation is there in spermatogenesis. Now let us see the differences between spermatogenesis and oogenesis. Okay. Spermatogenesis starts from puberty, but oogenesis from fetal life. That is first difference. Second difference, in spermatogenesis, one primary spermatocyte is producing the two secondary spermatocytes which are equal. But in oogenesis, one primary oocyte is producing two unequal cells, secondary oocyte and primary first polar body. Next, the meiosis also unequal in oogenesis, but equal in spermatogenesis. There is no differentiation in oogenesis, but there is differentiation occurs in the conversion of spermatid to sperm in spermatogenesis. Spermatogenesis occurs throughout life, whereas oogenesis will occur for a particular period only. It will be stopped at the age of 45 to 50 years, which is a phase called menopause. So these are the differences between spermatogenesis and oogenesis. And regarding oogenesis, let me give you the hormonal control. Same GnRH produced by the hypothalamus, it acts on the adenohypophysis, and adenohypophysis produces gonadotropins, FSH, LH. What is the duty of FSH in female? It causes the follicular growth like primary, secondary, tertiary, Graafian follicular growth. That is one function. FSH. What about LH? Two functions, guys. LH causes the rupture of Graafian follicle, helps in ovulation. After ovulation, corpus luteum is formed. That corpus luteum is maintained by the LH. And one ovum is produced in one menstrual cycle, but in in human ejaculates nearly 200 to 300 million sperms present. Sperms will be active for about 72 hours. But ovum, that means secondary oocyte, is viable for about 24 hours. 24 hours. So these are the points related to spermatogenesis and oogenesis. Now let us talk about menstrual cycle topic. The reproductive cycle in the female primates is called menstrual cycle, female primates. Primata is the order under Mammalia. It consists of monkeys, apes and humans. These are showing the menstrual cycle. Point number 1. The first menstruation begins at puberty and is called menarche. And cessation of menstrual cycles occur at the age of 50, that is called menopause. And in the menstrual cycle, there are events related to the ovary, there are events related to the uterus, both are present. That means ovarian cycle, uterine cycle put together called menstrual cycle. On an average, one menstrual cycle in a female occurs for about 28 to 29 days. For about 28 to 29 days. So all these cyclical events occurring in this particular period of one menstrual cycle till the next, one menstruation till the next menstruation is called menstrual cycle. We can divide the menstrual cycle into 4 phases for our convenience. Like first phase is called menstruation, followed by follicular phase, ovulatory phase, luteal phase. Let me give you the events of these phases one by one, listen carefully. Regarding the menstruation phase, it occurs for about 3 to 5 days. During this cycle, there is shedding of endometrium, and uterine endometrium comes out along with the blood, which is said to be menses. Menses. See this is the end of the cycle, this is the next cycle, menses. See what is happening to this red color line hormone, progesterone, estrogen, especially progesterone, there is a sharp decline in progesterone. So menstruation is due to the sharp decline in progesterone. Next, endometrium is required for the maintenance of pregnancy. So that means endometrium is shedding out, there is a menses, means there is no pregnancy. Absence of menstruation may indicate the pregnancy. It is not a certain indicator of pregnancy, it may indicate pregnancy, because the absence of menstruation can be caused by many other reasons. There are several reasons, like hormonal imbalance, like thyroxine can cause this. Next, diet, disease conditions, other physiological aspects can stop the menstruation. So that absence of menstruation is not a certain indicator, it may indicate the pregnancy. These are the points related to the menstruation. And when it comes to the follicular phase starts after menstruation phase. During follicular phase, the pituitary hormones FSH and LH gradually increases. Remember, first point, FSH, LH gradually increases. Regarding the FSH, I want to tell you, FSH is responsible for the follicular growth in the ovary, like primary follicle becomes secondary, tertiary, Graafian. And you already know that growing follicle secretes estrogen, so that estrogen also gradually increases during follicular phase. And estrogen is responsible for the regeneration of endometrium. See, endometrium thickness increases during follicular phase. These are the events of follicular phase. FSH, LH increases gradually, follicular growth, growing follicle secretes estrogens, estrogens causes the regeneration of endometrium. Of course, estrogens produced by the growing follicles, they are also required for the growth of the follicles. On the 14th day, on a typical menstrual cycle, FSH, LH and estrogen are peaks. Because of these three peaks on the 14th day, which is called ovulation day, all the three hormones are required for the ovulation, but main role is caused by the LH. The LH surge causes the rupturing of Graafian follicle, releasing the ovum in the form of secondary oocyte, along with other contents like first polar body, zona, corona and some granulosa cells. This event is called ovulation. After ovulation, the ruptured cells, remnants of the Graafian follicle, they are forming a temporary endocrine gland, corpus luteum. So after ovulation, the next phase is called luteal phase. During the luteal phase, what happens? Corpus luteum produces progesterone and estrogen. But progesterone is the main hormone. Progesterone peak should be slightly high compared to the estrogen. Mainly progesterone and estrogen. And progesterone maintains the endometrium. Estrogen is also required for the proliferation of thickness of endometrium. Hence, endometrium reaches the maximum thickness during a luteal phase. Progesterone maintains the endometrium. Okay, fine. The ovum that is released, if it is fertilized, what is the fate of the corpus luteum? If it is fertilized, that may lead to pregnancy. To maintain the pregnancy, corpus luteum continuously should produce the progesterone until the 3rd month of pregnancy, because 4th month onwards placenta produces progesterone. What happens if the ovum is not fertilized to the fate of the corpus luteum? If the ovum is not fertilized, by the end of the cycle, the corpus luteum degenerates and remains as a scare known as corpus albicans. When corpus luteum degenerates, that suddenly falls the progesterone and estrogen, causing the menstruation of next cycle. And what is happening to the FSH and LH levels during the luteal phase? Progesterone, estrogen are increasing, but FSH, LH are decreasing. The decrease in FSH, LH is because of progesterone, because progesterone inhibits the GnRH. It inhibits the GnRH. So when progesterone is inhibiting the GnRH, then FSH, LH reduces. That actually responsible it preventing the further growth of other follicles, because one follicle is ruptured, there are many other follicles, they may start their journey in the middle of the cycle itself. To stop that follicular growth further in this luteal phase, FSH, LH level should be decreased. That decrease is caused by the progesterone. See, there is a word called GnRH pulse frequency. See, in the follicular phase, high levels of estrogen compared to the progesterone it is having positive influence over the GnRH. It stimulates GnRH, so that FSH-LH increases during follicular phase. Next, estrogen levels compared to the progesterone, they are low, relatively low levels of estrogen. They will have negative influence over the GnRH. GnRH is decreased, thereby FSH-LH levels are decreased here. The follicular phase is called proliferative. Name follicular because of ovarian event called follicular growth. Name proliferative because of uterine event called proliferation of endometrium. The luteal phase is also called secretory phase. Luteal because of ovarian event because corpus luteum is formed. The secretory because progesterone maintains the secretions of endometrial glands. Hence it is called secretory phase. Whatever the number of days in the menstrual cycle, remember guys, the luteal phase is fixed, generally it is for 14 days. See, in a cycle, the first first 14, next 14. The end of the first phase last day is, the last day of the first phase is the ovulation day. Suppose there are 28 days in a cycle, minus luteal phase 14 days when you remove, the remaining days are the first 14 days. In the first 14 days, the last day is the ovulation day, the ovulation day is the 14th day. Based on this, you can say ovulation occur in the middle of the cycle. But if it is the 30-day cycle, minus 14, 16 days are there, last day will be the 16th day of ovulation. If it is 32 day, minus 14, it is around 18th day ovulation occurs. And once the ovum is fertilized with the zygote, if the female get pregnant, all the events of menstrual cycle will stop during pregnancy. They will be stopped. That is the important point I want you to remember. The next topic after menstrual cycle is fertilization. See, the site of fertilization is ampulla of fallopian tube. What are the events that are related to the fertilization? Let me explain. When the sperms are released into the vagina of a female, that is said to be insemination. After insemination, the sperms will undergo certain changes. Those changes that are happening in the sperm in the female reproductive tract is said to be capacitation. The capacitation is followed by, see, after capacitation the sperms will reach the fallopian tube. Simultaneously, if the ovum reaches the fallopian tube, see, fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. This is the reason why not all copulations lead to fertilization and pregnancy. So, once sperm and ovum reaches that area, the sperm undergoes certain changes called acrosomal reaction. Acrosomal reaction. So, what is acrosomal reaction? The acrosome of the sperm releases certain enzymes required for fertilization. Those enzymes helps the sperm to cross the corona radiata, to cross zona pellucida and enter into the space called perivitelline space between zona pellucida and the membrane of the ovum. And finally, the sperm after acrosomal reaction, it will enter into the cytoplasm of the ovum. That is the sperm oocyte membrane fusion. So, first acrosomal reaction followed by the entry of sperm into the cytoplasm of the secondary oocyte. Once sperm enter into the cytoplasm of the secondary oocyte, now the membrane of the plasma membrane of the secondary oocyte undergo depolarization, which is called fast block for the entry of sperms, followed by a reaction known as a cortical reaction. The cortical granules present in the cytoplasm of ovum, by exocytosis they will go to the zona and they will block the receptors present on the zona for sperms. They are called ZP3 receptors, they are blocked. So, the cortical reaction leads to zona reaction. During zona reaction, the entry of sperms will be blocked permanently. That is called slow block, which is responsible for preventing the polyspermy, entry of many sperms. That is the reason why only one sperm can enter the secondary oocyte of the one sperm can enter into the secondary oocyte stage of the ovum. So, cortical reaction followed by zona, then the secondary oocyte completes meiosis 2. Already secondary oocyte is there, first polar body is there. Once the sperm enter, secondary oocyte undergo division and form oocyte and second polar body. So, unless and until the sperm enter the secondary oocyte will not complete the meiosis 2. Once meiosis 2 is completed, oocyte, which is also called ovum, is produced. then second polar body is produced. After this, the nucleus of sperm fused with the nucleus of ovum, pronuclear fusion, which is called amphimixis. Amphimixis results in the formation of a zygote. That will be the completion of fertilization process. The fertilization occurs in the ampulla region of fallopian tube. Once the fertilization is completed, now the zygote start divisions. The divisions of the zygote is said to be cleavages. See this? The divisions of the zygote are cleavages. The zygote, immediately after fertilization, corona radiata disappear. While it is passing through the fallopian tube, the zygote undergo division. The first division, first cleavage of the zygote occurs while it is passing through the isthmus. It undergo one division and forms the two cells. They are called the two blastomeres. Two blastomeres divide to form 4 blastomeres. 4 divide to form 8 blastomeres. The 8 to 16 blastomere stage embryo is known as morula. See this 8-celled morula is there at this junction, but first division occurs while it is passing through the isthmus. Then morula is also called mulberry. Still there is a zona pellucida around morula. There are cell junctions between the cells of the morula, 8 to 16. Then there is a cavity formed in the morula inside. Cavity is formed, lifting the cells to one end of the morula, forming a layer around the morula. This is called trophoblast layer, made up of trophoblast cells, and the remaining cells of the blastocyst are called inner cell mass. Inner cell mass, and this is called the cavity. Development of cavity inside the embryo is called cavitation. So with this cavity, trophoblast and inner cell mass, this is called blastocyst. The blastocyst is the stage meant for implantation. And once the blastocyst is formed, the zona pellucida will immediately disappear. Now this blastocyst is ready for implantation. So what is the site of implantation? Uterus. So implantation starts the 6th day after fertilization. It will be completed by the end of the 2nd week, either 13th or 14th day. So during implantation, the trophoblast cells produces some proteolytic enzymes. So this is the endometrium of the uterus, and this is the embryo that is going to implant. So with the help of trophoblast cells, by producing proteolytic enzymes, the embryo will invade the endometrium until the maternal blood vessels get reached and breached. This step is called implantation. And during the implantation, the trophoblast cells will divide and form trophoblastic villi. Later, they will be converted into the placental villi, nothing but chorionic villi. Next, during the implantation itself, the endometrial cells will divide and surround the embryo. That is called decidua. So decidua is to protect the embryo. So cells of endometrium that are dividing and surrounding the embryo is called decidua. So this is called implantation. Once implantation is done, it can be known as a pregnancy. Once the implantation is done, now the placenta formation starts. So placenta formation will be completed by the end of the 12th week. That means by the end of the 3rd month. So how the placenta is formed? What are the structures that are involved in the formation of placenta? See this is the placenta. The placenta is formed by the endometrium of the mother along with the capillaries of mother with the fetal capillaries, which are also called chorionic villi. So the placenta is a structural and functional connection formed by the maternal tissues and fetal tissues. Maternal tissues are endometrium plus mother capillaries. Fetal tissues are fetal capillaries, nothing but chorionic villi formed from the trophoblastic villi. And placenta is connected to the fetus through umbilical cord. Umbilical cord consists of blood vessels like umbilical arteries and umbilical veins. See this? From the umbilical arteries, a pair of umbilical arteries, the blood of fetus will go to this placenta area. And there are several capillaries fetal capillaries surrounded by the maternal blood. So with at the capillaries, from the maternal blood into the fetal capillaries, from fetal capillaries into the maternal capillaries, exchange of materials occur. Nutrients from mother to fetus, oxygen from mother to fetus. After that exchange, blood, nutrient rich oxygenated blood will be returned to the fetus through umbilical vein, one umbilical vein. So, umbilical cord is the connection connecting the maternal tissues with the fetal tissues. And 100% fetal blood only passes through the umbilical cord, a pair of umbilical arteries and a umbilical vein. Umbilical artery carries deoxygenated blood, umbilical vein carries oxygenated blood here. So, the placenta is the structural and functional connection, and what are the functions of placenta? The first and foremost function of the placenta is it is involved in the exchange of materials between mother and fetus, exchange of nutrients, waste, hormones, gases like oxygen and carbon dioxide. The second function is placenta secrete hormones. There are 5 hormones, important hormones are secreted by the placenta. One is HCG, second one is HPL, third is relaxin, fourth estrogen, fifth progesterone. So, these are the 5 hormones that are secreted by the placenta. What is the function of HCG? HCG, it maintain corpus luteum during pregnancy, and during pregnancy LH levels are absent in the pregnant female. Actually LH maintains corpus luteum, but who is taking the function of LH during pregnancy? HCG maintains corpus luteum for the maintenance of pregnancy. When corpus luteum is maintained, you know, corpus luteum produces progesterone. Progesterone maintains pregnancy, but the same progesterone inhibits GnRH, thereby LH will be inhibited during pregnancy. HPL, human placental lactogen, also called somatomammotropin, it has anti-insulin effect. That means it will not inhibit the release of insulin, but its effect is against insulin because it increases the blood glucose levels of the mother for more glucose availability for both maternal and fetal tissues. Next, relaxin. Relaxin relaxes the muscles of the uterus for the maintenance of pregnancy. Estrogen, progesterone functions we already know. Estrogen along with the progesterone maintain pregnancy required for the growth of the uterus, that means a thickening of the endometrium. Progesterone maintains pregnancy by preventing the contractions of uterus, also causes the differentiation of alveoli of mammary glands and helps in lactation, milk secretion. So, these are the functions of hormones of placenta. Placenta is having other functions like placenta produces immunosuppressors because mother produces antibodies against the father chromosomes of this fetal cell, paternal chromosomes, and by producing immunosuppressors it suppresses the immune response, and it also protects the fetus from the high blood pressure of the mother. So, these are the general functions of placenta. Human placenta is called discoidal, like a round plate like structure. Human placenta is called hemochorial because the fetal capillaries are bathed in the mother blood, and human placenta is said to be chorioallantoic. So, it is formed from the capillaries of both the chorion and allantois. Next, the fetus also develops extraembryonic membranes around it. What are the extraembryonic membranes present around the fetus? The extraembryonic membranes include amnion, see, if it is the fetus, the immediate membrane is amnion. Between fetus and amnion there is amniotic fluid. Amniotic fluid acts as a shock absorber. There is an extraembryonic membrane called allantois, extraembryonic membrane called yolk sac, surrounded by chorion. Chorion actually forming the chorionic villi, responsible for the formation of placenta between the mother and fetus. So, the functions of layers like amnion acts as a shock absorber with amniotic fluid and protects the fetus. Yolk sac involved in the production of RBC, red blood cells. Allantois involved in the storage of waste, and chorion responsible for the formation of chorionic villi, and chorion is the layer covering all the extraembryonic membranes. So, this is about placenta and extraembryonic membranes related to the fetus. Once the embryo gets implanted, the inner cell mass present in the blastocyst of the embryo, which is also known as embryo proper, it has stem cells. Now, it will develop into the complete embryo. The stem cells present in the inner cell mass divide to form 3 germinal layers. They are ectoderm, endoderm and mesoderm. 3 germinal layers, ecto, endo and meso. And these 3 germinal layers are involved in the formation of almost all the tissues and organs of the body. So, inner cell mass, after implantation, the stem cells of the inner cell mass are forming germinal layers are forming all the tissues and organs. Next, if you see the stages in the development, first is the zygote. Zygote forms a stage called morula. Morula forms a stage called blastocyst. Then there is a process called gastrulation for the arrangement of cells and forming 3 germinal layers like ecto, endo, meso. After gastrula, then next is fetus. So, till the 9th month we can call it as embryo, from 9th month onwards it is fetus. And see the gestation of embryonic events that are occurring during the embryonic development. We can divide the gestation period of humans into 3 trimesters because it is around 9 months. First trimester 1 to 3 months, second trimester 4, 5, 6, third trimester 7, 8, 9 months, around 9 months. Gestation of other animals also they have given, there was like dogs around 60 to 65 days, cats 58 to 65 days and elephants around 2 years 607 to 641 days. And during a pregnancy, what happens regarding the developmental events of the fetus? See, in the first trimester after the 1 month of pregnancy, the embryo's heart is formed. So, the first sign of growing fetus may be noticed by listening the heart sound carefully through the stethoscope. Next, by the end of the 2nd month of pregnancy, limbs and digits are formed. And by the end of the 3rd month of first trimester, along with the limbs, digits, external genitalia also formed. Overall first trimester is meant for major organogenesis. When it comes to the 2nd trimester, during the 5th month of 2nd trimester, during the 5th month, see the first movements of the fetus can be seen during 5th month. Appearance of hair on the head are usually observed during 5th month. By the end of the 2nd trimester, that means by the end of the 24th week, end of the 2nd trimester, the body is covered with fine hair, eyelids are separate and eyelashes are formed. So, remember these events: after 1 month heart, after 2 months limbs, digits, after the 3rd month limbs, digits, external genitalia, during 5th movements plus hair on the head. By the end of the 6th month, 12th week, eyelid separation, eyelashes formation and body is covered by fine hair. And by the end of the 9 months of pregnancy, the fetus is fully developed and is ready for delivery. That process is called parturition. Let me tell you the events related to the parturition. So, parturition is a neuroendocrine mechanism. So, regarding the events of parturition, the first one is fully grown fetus and placenta, they induce signals required for fetal ejection reflex. So, who will induce the signals for fetal ejection reflex? Fully grown fetus and placenta. What is fetal ejection reflex? Mild contractions of the uterus. Mild contractions of the uterus. When generally contractions are prevented so far because of progesterone, but by the end of the pregnancy, compared to the estrogen levels, progesterone levels get decreased. So, that causes mild contractions. Those mild contraction events is said to be fetal ejection reflex. This fetal ejection reflex mild contraction signal will go to the brain of the mother, and hypothalamus produces oxytocin, and that will come to the uterus and increases the strength of the contraction. See, fetal ejection reflex is mild contraction that induces the release of oxytocin. Oxytocin increases the strength of the contractions. That wild contractions is said to be labor, and that labor causes the pushing of the child out of the birth canal by a process called parturition, childbirth. And during parturition, the ejection of fetus, placenta, decidua, extraembryonic membranes can occur. All these contents comes out by a process called parturition. And I told you it is a neuroendocrinal mechanism because it has the role of nervous system as well as hormones. What are the hormones that are involved in this neuroendocrinal mechanism? The hormones like estrogen, progesterone levels get decreased, oxytocin, cortisol, prostaglandins. These are involved in parturition reflexes. Remember, oxytocin, estrogen, cortisol, prostaglandins are involved in parturition. And compared to the estrogen, the progesterone levels will decrease by the end of the pregnancy. And what are the hormones that are increased during pregnancy? Let me give you this last point. During pregnancy, the hormones are increased in manifold. They include estrogen, progesterone, prolactin, cortisol, thyroxine. So, what are the 5 hormones that are increased manifold during pregnancy as per our syllabus? Estrogen, progesterone, prolactin, cortisol, thyroxine. Functions of those hormones we already know, but I want to give you it again. Estrogen, progesterone maintains pregnancy. For the growth of endometrium, maintenance of endometrium, these hormones are required. Prolactin is required for the differentiation, growth of the mammary glands and milk secretion. Cortisol increases the blood glucose levels and maintains the availability of glucose, and also cortisol is required for the maturation of lungs in the fetus, and cortisol also prepares the mother for the delivery by the end of the pregnancy. About thyroxine, thyroxine maintains the metabolic changes in the mother. So, overall functions of these 3 hormones are given like these 5 hormones increases in manifold to maintain the metabolic changes in the mother, to support the pregnancy, to support the fetal growth. These are the functions of these 5 hormones that are increased manifold during pregnancy, and parturition is followed by lactation. But remember, milk secretion starts by the end of the pregnancy itself. It will be increased after delivery while feeding the young. Okay. So, regarding the lactation, the average duration of human pregnancy. Mammary glands of the female undergo differentiation during pregnancy and starts producing milk towards the end of the pregnancy by a process called lactation. So, which hormones are responsible for lactation? Progesterone causes lactation, but after delivery prolactin is responsible for lactation, because after delivery as the placenta get ruptured and it is coming out, progesterone levels are absent in the blood of a female. So, that after delivery the hormone that is mainly responsible for lactation is prolactin. Okay. This helps the mother in feeding the newborn. The milk produced during the initial few days of lactation is called colostrum. Colostrum consists of several antibodies. The antibodies like IgA, absolutely essential to develop resistance for the newborn babies, and also consists of some essential nutrients which are required for the growth of the baby. And breastfeeding during the initial period of infant growth is recommended by doctors for bringing up a healthy baby. So, what are the antibodies present in the colostrum? Important antibodies present in the colostrum are IgA. More amount of IgA antibodies can be seen. So, with this the human reproduction topic including all the events covering all the topics is completed. Thank you.